The outer layer of the eyeball, the fibrous tunic made up of collagen and elastic fibers. These little folds are just in the preparation of the slide. You get a little fold in the tissue and you don't want to pull it too hard. So, but it usually looks pinkish. Inside, moving, that's the outside of the eyeball. We actually have, this is cross-section from skeletal muscle. Remember, the muscles of the eye insert on the sclera. And so that's actually skeletal muscle. The next layer in then is the vascular tunic. Vascular tunic made up in this area of the choroid. If we would go forward, we find the ciliary body and then the iris, but we're in the back 4/5 of the eyeball. We have pigments that we find are pigments and blood vessels. Then the next little line that you see right here is the pigmented layer of the retina, a single layer of cells. Embedded in it are the rods and cones. The rods and cones extend this far. They're cells where the nucleus is about in the middle of it, but it's long cell. So that's the nucleus of the rods and cones, but the rods and cones are this layer right here. They synapse with the bipolar cells. The bipolar cells, lots of nuclei, but this is the bipolar cells. That's just the nuclei of them. And then the innermost layer are the ganglion cells, and here's the nuclei of the ganglion cells, and their axons then come back and form the optic nerve. This slide doesn't have the optic nerve, but on the dissecting scope, you'll be able to see the optic nerve and what is the blind spot. So let's look at this at one higher magnification. This is usually what I do on a lab practical. And I was just lucky there weren't any tears in this. Usually there's tears. This is just a really, really nice slide. Sclera, choroid, pigmented layer of the retina, rods and cones, bipolar layer, ganglion layer. If I move, I'll bet if I move closer to the back of the eyeball, more and more ganglion cells, because we have less and less convergence and more resolution. This has to do, just think about the receptive field. Small, it's not really what it is, but whatever, it gives the same effect. If I move further away, I'll get fewer and fewer ganglion cells, more convergence of the information. That's why peripherally, your vision's a little blurrier. All right, any questions on this one? That's a great slide.